So our next speaker is Dr. David Markowitz, and he works for IARPA, and his role is to explore very high-risk, high-reward technologies to advance engineering, science, and products. The title of the talk is Biology is All You Need. It's a nice follow-on to the requirements created by the previous speaker, potentially some interesting, exciting solutions. Dr. David Markowitz. Thanks.
Biology is all you need. My name is David Markowitz. I'm a biotechnologist and a computational scientist at IARPA, where I run a large portfolio of high-risk, high-reward research programs at the interface between biology and computing. IARPA's mission is to deliver transformational capabilities to the national security community. As you can imagine, a major focus of our work is on next-generation computing technologies, such as quantum computing, cryogenic computing, and neuromorphic computing. And I'm here today to show you that the key to addressing some of our most urgent scaling challenges in data storage and computing may come from an unlikely source: biology. I'll be talking about two topics that are going to sound like science fiction: storing exabytes of data in DNA and reverse engineering neural circuits in the brain to guide next-generation computing architecture development. Even if you're skeptical that biology has any value to offer for computing, stick with me, because I think you'll be inspired by what's now possible at the interface between biology and engineering. So let's begin by talking about large-scale data storage. There is a looming crisis in the data storage field. The world creates and consumes exponentially more data each year, 90 zettabytes alone in 2022. By contrast, the global supply of data storage can't keep up. We have barely enough installed storage capacity today to store 5% of the globally produced data. This is a problem for government intelligence analysts, who often need to sift through mountains of historical data to understand why a world event happened and who was involved. If we can't store historical data, we can't do digital forensics, and national security suffers. This is also a problem for industry. Tech companies need to store mountains of data, not just our personal photos and videos, but the reams of transaction data that power online commerce. If we can't store enough data, the digital economy suffers. We are in urgent need of a fundamentally new solution to address this scaling crisis. Historically, our solution to this problem has been to build ever larger data centers. These are football field-sized facilities that consume megawatts of power and cost massive sums of money to build, operate, and maintain. For those of you who aren't used to thinking about data storage at this scale, an exabyte is 1,000,000 terabytes. That's roughly the scale of the largest commercial data centers today. And currently, the 5-year total cost of ownership of a 1 exabyte data archive is in the hundreds of millions of dollars. And that's to maintain one copy of a large data archive that you don't access very often. If you want a backup, that's another $100 million. And all of this is to keep just a tiny fraction of the data that we create. Scaling up a data archive to store over a zettabyte, which is 1,000 exabytes, would cost over $100 billion. Again, as you can see, there is a pressing need for innovation in data storage to enable larger scale at a much lower price point. Today's data storage media are expensive to use for two reasons: data density and data stability. Traditional planar media, such as magnetic tape, DVD, and hard disk, write data in 2 dimensions. And once you've maximized your aerial storage density in 2D, if you want to store exponentially more data, you need to manufacture exponentially more media and the devices for interfacing with those media. And that's a major driver of cost when we are scaling data centers. Ideally, we would like a storage medium that scales gracefully in 3 dimensions, so that when we want to store exponentially more data, we don't have to spend exponentially more money. The other major driver of cost in today's data centers is something called bit rot, or the passive degradation of information over time. All of the storage paradigms that are in use today suffer from this problem. And this is a major driver of the need for routine data integrity checks and media replacement, which is a major cost driver. Ideally, we would like a storage medium that is so stable it doesn't degrade over time. We can just write it and forget about it. Thankfully, biology has already solved this problem. Living systems need to store genetic information in a medium that's extremely dense and extremely stable. DNA is that medium. You can think of DNA as a molecular scale sequence of physical bits. The bases in DNA, A, T, G, and C, are nanometer scale bits. And because DNA is a three-dimensional molecule, it can be coiled and compressed to achieve extremely high volumetric information density. It's been estimated that DNA can store information with a density that is fully 5 orders of magnitude higher than any man-made storage medium, including 3D NAND. And here I'm showing an electron micrograph of the entire genome of a mammalian cell that's compressed into a micron-sized space in a nucleus. In principle, DNA is so dense that we could store exabytes of data in a size equivalent to a ping pong ball. DNA is also incredibly stable. Biologists have sequenced the genome of a 700,000-year-old horse because under the right conditions, the chemical bonds that hold DNA together don't degrade over time. If we can exploit the density and the stability properties of DNA for enterprise storage applications, it would be a game changer for the industry. For those of you who don't know, DNA data storage is actually a pretty vibrant research field, as well as a nascent industry, which I'll be telling you about in a moment. The steps involved in storing and retrieving information from DNA are pretty straightforward. I'm showing you a diagram here. We start with binary data that encode files that we would like to write. Using a special codec, we convert binary to the letters that make up DNA, A, T, G, and C. And then we use special DNA synthesis technologies to grow new strands of DNA that encode those sequence of letters that we encoded in the previous step. We can then collect that into ultra-dense droplets that are very tiny and store that for arbitrary amounts of time, 10 years, even 100 years. When we want to retrieve information, we retrieve the droplet that's of interest, pop the DNA into a sequencer and read out the sequence of letters. And then in the final step, we can use our codec to convert the letters of DNA back to binary. To date, the largest published DNA data archive is about 200 megabytes. This was a milestone achievement for this research field, besting previous efforts by at least an order of magnitude. But for archival storage applications at the enterprise scale, this isn't quite enough to be compelling, to show that this technology is ready for prime time. And the reasons for this, that we're still operating at this scale, is because of the cost and the throughput of DNA synthesis and sequencing technologies. Using today's synthesis approaches, it costs $100,000 to write a gigabyte of data to DNA, and about $500 per gigabyte to read it back out. Clearly, this is not economical for large-scale enterprise data storage, especially when you want to move to the exabyte regime. At IARPA, I helped design and launch the Molecular Information Storage, or MIST program, to overcome these and other challenges. This is a moonshot effort to deliver revolutionary capabilities by bridging disciplines, in this case, the tools of the semiconductor and biotechnology industries. Our goal at IARPA is, by 2025, to make DNA data storage scalable, economical, and practical for enterprise archival use. And I've put some of our specific quantitative targets up on this slide. We're moving from systems that can only write about 20 megabytes of data to DNA in a single run today to systems that can write a terabyte of data per run. We're dropping the cost of DNA synthesis from $100,000 per gigabyte to $1 per gigabyte. And we want to demonstrate all of this on an end-to-end workflow that works on a tabletop. These will be about 4 orders of magnitude improvements in scale and reduction in cost relative to conventional technologies. The reason we've set these targets is because this will present a compelling de-risking of this technology stack for industry and will empower industry to begin contemplating the development of commercial products around DNA. The vision of the MIST program is to make it possible to shrink an exabyte-scale data center down to a tabletop form factor, something that you can just plug into a wall outlet, write your data quickly, easily, and forget about it, with the additional benefit of scalability beyond the capabilities of today's storage paradigms. DNA data storage might sound like science fiction, but I'm here today to show you that because of work by IARPA and our partners in this space over the last several years, it's becoming science fact. Here I am holding, if we could have this up on the screen, our next generation DNA synthesis chip that uses integrated CMOS to control the synthesis of 128 million strands of DNA in parallel. This is enough to write about 2 gigabytes of data on a chip, which you can then easily collect into a droplet for long-term storage. All of the control electronics, if you can cut back to the slide, all of the control electronics for operating this chip comfortably fits on a tabletop, and it only costs about $100 per run. That is a several order of magnitude improvement on the state of the art from when we started this program. Furthermore, because we're using the tools of the semiconductor industry, we are now on an exponential scaling roadmap that will make it possible for us to produce chips that can write a terabyte to DNA in a single run, and ultimately, once there are commercial appliances that are ready for data centers, you'll be able to write a petabyte per system in a short amount of time, and eventually we have a clear path to scaling into the exabyte regime. This technology is real, and it's becoming commercially viable. Now, this chip was developed by a team from the Georgia Tech Research Institute and Twist Bioscience under an IARPA MIST contract. This represents a new category of microfabricated device that uses onboard CMOS electronics to control the synthesis of millions of DNA oligomers in parallel. Previously, this was all done with inkjet printing at fairly low density, but we can use the tools of the semiconductor industry to fabricate millions and eventually billions of self-contained synthesis devices on a chip. We can use these devices to control the acid concentration or the pH in solution in very small synthesis zones on the chip, as shown here. And this allows us to electrochemically control reactions that are involved in DNA synthesis. This is a fully automated approach that uses minimal power and chemical reagents for a very low operating cost. And if you make enough of these chips during a fab run, the cost per chip becomes quite low as well, potentially just $1 or $2. Now, the MIST program developed multiple distinct synthesis platforms that use this general approach. Our second platform was developed by a team from the Broad Institute, the Harvard Center for Nanoscale Systems, and DNAscript. This team published the details of their chip design and synthesis approach in the summer of 2022, as shown here, so you can read all about it. Together, these platforms show that IARPA has cultivated an ecosystem of multiple new DNA synthesis technologies that can support data storage in the future. IARPA has also invested in multiple sequencing platforms that can further improve the scale and reduce the cost of data readout from DNA. One of our approaches adapts an existing commercial instrument to support DNA data storage. This is an Illumina MiSeq DNA sequencer behind me that's been modified to read spatially structured information directly from our DNA synthesis chips. And this allows us to read data with orders of magnitude higher throughput and lower cost than conventional DNA sequencing for life sciences applications. IARPA has also funded an early-stage commercial platform that's designed to work with droplet-based archives. This is a CMOS chip from a startup called Roswell Biotechnologies that has the potential to enable very high throughput DNA sequencing for storage applications using a different technology. Now, these are just two options for DNA readout, however. There's a rapidly growing ecosystem of sequencing technologies that can be used to support data storage applications. It's important to note that DNA synthesis and sequencing methods have some unique error modes relative to existing data storage paradigms. For any given DNA sequence, We have to contend with base substitutions, insertions, and deletions as shown here, such that the per base error rate might be 5% overall. We also need robustness to erasures of whole DNA sequences, which might not get synthesized in the first place, or may get lost during sequencing. This may sound daunting, but we can overcome these problems by adapting error-correcting codes from the storage industry. DNA storage requires a coding framework that's matched to the storage media and is robust to the error modes of the write and the read channels. IARPA and our partners on the MIST program at Los Alamos National Laboratory have developed a novel codec that uses Reed-Solomon erasure coding to allow for tunable robustness to any channel error model for DNA storage. This is an open source tool. You can go to the GitHub page and review the documentation and use it for yourself today. The basic idea here is, with DNA data storage, we write sequences of letters called oligos that are typically about 150 bases long, and we write millions or billions of those sequences in parallel. In the middle of each one of those oligos is a data payload. The ADS codec that was developed by Lannel encodes blocks of data in that data payload. Each block has data and a parity bit, and then an associated metadata block that encodes an address and an erasure flag. At the outer codec level, we rotate these blocks across oligos so that there's some redundancy across the archive. And we further use erasure oligos to confer robustness to the loss of entire oligos during synthesis or sequencing. I want to mention that designing DNA codecs is very much a research problem, and this is an area where storage experts can still have a huge impact on this developing field. Now, as I said earlier, the goal of the MIST program is to develop a complete technology stack that makes DNA data storage and retrieval cheap, scalable, and practically useful for the first time. And I'm happy to share that this program has catalyzed the growth of a new industry. After IARPA awarded MIST contracts in 2019, several of our teams founded the DNA Data Storage Alliance to roadmap this new industry, to establish interoperability standards for commercial products, and to identify how they were going to best meet the needs of initial commercial customers. This organization now has over 50 members drawn from the tech industry, the biotech industry, the traditional data storage industry, and a large number of academic institutions and nonprofits. Collectively, this ecosystem is spending hundreds of millions of dollars to roadmap commercial products, and that is separate from the US government's investments in this space. They are working to develop commercial products that can be installed in data centers within the next 2 to 3 years. And we believe there will be demand for archival DNA storage appliances in data centers once you look at the economics of data storage at large scale. In this bar chart, the yellow bars in the middle of each plot show the cost of DNA archival storage once we can get the cost of synthesis down to about $100 per terabyte. And as you can see here, once we hit that price point, DNA storage becomes more economically attractive for long-term, 10-year-plus archival storage than existing paradigms, magnetic tape and archival storage in the cloud using glacier approaches. So we expect there will be demand for this once we can achieve that particular price point. To conclude this part of the talk, it's been said that innovation happens at the interface between disciplines, and DNA data storage provides a case study of exactly that. IARPA deconstructed the pain points of enterprise data storage and determined that biology had already solved these problems. We invested in bridging the semiconductor and biotechnology industries to produce devices that will allow us to write and read DNA at scale and for low cost. And now we have a rapidly maturing industry. And IARPA expects that within the next few years, we're going to see the first archival DNA storage appliances in data centers. And eventually, this will empower all of humanity to keep all of the world's data cheaply and compactly in DNA. Now let's switch gears and talk about how biology can help us solve a foundational problem in computing. I talked about the storage crisis in part 1, and now I'm going to talk about a looming crisis in the field of AI. We have seen an impressive improvement in the capabilities of artificial intelligence systems over the last decade. And the leading edge of these advances are the largest models, which have been growing exponentially in size. In 2022, the largest model was a language model with 540 billion parameters. The cost of training this can be measured in billions of petaflops. The problem that we face are that the resource requirements for training our largest AI models have become truly extreme. This 500 billion parameter model required 6,000 custom processors running in a data center with megawatts of power. If you were to it's been estimated that if you were to train this model in the commercial cloud, which is broadly available to everybody, it would cost on the order of $10 million to train this model. And the carbon footprint would be equivalent to about 200 US homes per year. Now, given the massive growth in the size of these AI systems over the last 10 years, it's reasonable to contemplate a future where we want to train models that are fully 1,000 times larger than today's best systems. And this is where things really start to break down. To train a model with 100 trillion parameters in the commercial cloud would cost at least $10 billion and would have a carbon footprint equivalent to a medium-sized US city, about 200,000 homes per year. Even with the benefit of AI accelerators that are on the horizon to bend this curve, we're still headed toward a brick wall. And this isn't even a hypothetical future problem. We are already hitting the wall. There are several recent articles from 2022 that indicate power is already a bottleneck for data centers. In the United States and Europe, there just isn't enough power to build out new data center capacity. And where there is power, it's forcing difficult trade-offs between building new housing or new data centers. So the idea of scaling up compute capabilities by several orders of magnitude to meet growing AI needs is plausible, at least using today's paradigm. Of course, biology has already solved the power and scaling problems that I highlighted earlier. Our brains are proof by existence that efficient real-time training and inference is possible on models with 100 trillion parameters. Computation and communication in the brain consumes between 10 and 20 watts of power, judging by metabolic demands. And if you run the math, the cost of continuously training a human brain for 24 hours a day for the first 18 years of life is only about equivalent to about a third of a US home per year. This is proof by existence that general intelligence can be implemented and efficiently. So how do we bridge this performance gap? Neuromorphic computing hardware gets us closer to the efficiency of brains. Over the last 10 years, we've really seen a golden age of neuromorphic computing where there has been a steady march of improving capabilities using hardware platforms that use computing principles that are inspired by the brain. Event-based communication or spike-based communication, parallel sparse computing principles, processing in memory. The general idea is to minimize the amount of data that needs to be moved around on the chip. And indeed, when used as deep neural network inference accelerators, neuromorphic chips can be more than 100 times more power efficient than GPUs. But we still need more than 1 million times better power efficiency to rival biology. IARPA's hypothesis is that neuromorphic platforms still lack key guidance from neuroscience that's needed to implement truly efficient computing models. Specifically, we need a deeper understanding of how the brain efficiently represents data, transforms data, and learns from data. That's the key to unlocking the potential of neuromorphic hardware and allowing us to scale AI systems in the future. In 2016, I helped design and launch the IARPA Machine Intelligence from Cortical Networks, or MICRONS program, specifically to provide us with the guidance needed to realize neural computing systems with brain-like performance. And we took a very direct approach. By developing next-generation neuroscience tools needed to map complete synaptic connectivity and activity of hundreds of thousands of neurons spanning multiple regions of the mammalian brain, spanning a cubic millimeter of tissue. Now that may sound small, but it's large enough to encompass 4 different regions of a mouse brain, which I'll tell you about in a moment. Our goal was to distill these data into computing primitives that can guide algorithm and architecture development. I'm going to tell you briefly about the experimental data acquisition portion of this program, and then we'll dive into some really exciting advances that are providing guidance for algorithm and architecture development. So the first step of this program was to functionally image the activity of as many neurons as possible in an awake behaving mouse brain. This was a partnership with Andreas Tolias's group at the Baylor College of Medicine. So we developed very large-scale functional imaging approaches that allow us to look at about 100,000 neurons blinking on and off while an awake animal is shown a wide variety of visual stimuli. And in this plot in the middle here, I'm showing you the delineation between 4 separate regions of early mouse visual cortex that we functionally imaged. V1 is primary visual cortex, and the lettered areas are higher areas. So this is analogous to looking at multiple layers of a deep neural network simultaneously. We can look across all of the depths of the cortical sheet. So mammalian cortex has 6 layers. We can look at all of them. And we can monitor the responses of these neurons for hours on end while we explore the broadest possible response, stimulus and response space for these neurons. The next step was to send the functionally imaged tissue off to the Allen Institute for Brain Science in Seattle, Washington, where this tissue was serially sectioned with a kind of scientific slicer that prepared 40 nanometer thin sections out onto a movie reel called grid tape. And then using a novel electron microscopy technology that was developed by the Microns program, the Allen Institute did very large field of view electron microscopy imaging of each of these sections. So we have 4 nanometer pixel resolution, and we're looking at sections that span millimeter fields of view. Each one of these images is about 125 gigabytes of data, and I'll give you the full view of one of them in a moment. The next step, after making about 25,000 of these sections, is to align them to each other to recover the full 3D volume that we had functionally imaged and then structurally imaged. So this was work, again, by the Allen Institute and by Sebastian Sung's group at Princeton University. After recovering the 3D structure of this block, now it's time to map the connectome, which is the synaptic connectivity between all of the neurons in this volume. For this, we use computer vision. This is a fully automated approach. And the basic principle is paint by numbers. You trace the outline of each stained cell body or neurite in each 2-dimensional section, and then we agglomerate them across sections to recover the 3D morphology of cells. We also use computer vision to identify the locations of synapses, of which there are about 500 million in this cubic millimeter volume. When we started this program, our first pilot data set encompassed the cell bodies of about 300 neurons. And this itself was 20 times larger than the previous state of the art. We then scaled that up by a further 400x to generate these 125 gigabyte electron microscopy images that cover millimeter extents and encompass thousands of neurons in one field of view. After you acquire about 25,000 of these images, it's enough to cover a cubic millimeter of tissue, and it's about 1.4 petabytes of data. This was, and I believe remains, the largest single biomedical imaging data set ever acquired. And this is enough to image 200,000 cells, and it fully encompasses the roughly 80,000 neurons that we functionally imaged in the steps that I showed you earlier. And just for a relative sense of scale, this is still 400 times smaller than the complete mouse brain, but it's enough for us to look at the interactions across multiple stages of the cortical hierarchy. So we have scaled up imaging capabilities by more than 1,000x on the Microns program to give us a new window into computation in the brain. The automated segmentation and circuit mapping approach that we used on this program is called chunk flow. There's a pointer to the paper on the archive here. I'm showing some of the steps from chunk flow here to highlight that this is a high-performance computing problem. When you use automated computer vision techniques applied to a petabyte scale data set, it's a truly massive computational burden. So to process the microns data required 3,000 GPUs running full bore for several days at a time, and it produced several petabytes of intermediate data. This is an example of how we are using high-performance computing to drive advances in neuroscience so that we can use neuroscience to drive further advances in high-performance computing. It's a virtuous cycle that we're creating on the microns program. Once you have automatically segmented each of these images, this is what it looks like. And I'm showing you a couple of detailed views to give you a sense for the millions of processes that need to be traced in just one of these images. After you agglomerate all of the traced sections to recover the 3D structure of neurons, this is what you get. The microns program has recovered the very first complete reconstructions of cells in the cortex of any animal. And when I say complete, I mean we've got the complete dendritic trees, which were truncated in some other previous data sets through other research efforts. We have the complete axonal arbors that span over millimeter scale distances. And so we now have the ability to densely map all connections in this volume of tissue. As this rendering shows, the IARPA microns program has achieved one of the holy grails of modern neuroscience, which is to fully map cortical columns. Here we're looking at a selection of 200 excitatory neurons that span all layers of the cortical sheet. You can see axons exiting the cortex at the bottom and dendrites extending all the way to the top, up to the cortical surface. This is an exciting achievement for biology, but it's also an important milestone for computer science because now we have the detailed structural and functional guidance needed to understand how the brain achieves such vastly superior performance to today's low-power computing systems. Now, these advances will only happen if people use the data. And it was a priority for IARPA as soon as we produced this massive data set about a year ago to make it freely available to everyone. And through the generosity of several cloud providers, Google Cloud and Amazon Web Services, these data are now freely available as public data sets. And if you go to microns-explorer.org and click that Explore button, you can bring up this entire data set for free in your web browser, look at 3D reconstructions, track synaptic interactions between cells. It's really extraordinary. We also have an API that allows researchers to programmatically interact with the data to do more nuanced analysis of structure-function relationships. So what can we do with these data? Well, one of the most fundamental questions we would like to answer is, what learning algorithm does the brain use? The prediction made by most of today's learning models is that the brain should use graded analog synapses. If you're using backpropagation with gradient descent to train a deep neural network, that produces an analog distribution of synapse strengths. When we look at microns data, that's not what we see. If you look at excitatory synapses in cortex, right? So we have these 3D reconstructions of the morphology of cells. We can look at the postsynaptic sites, the spine heads that excitatory neurons synapse onto. And it's well established in neuroscience that the volume of these spine heads correlates very highly with the strength of a synapse in the brain. So when we histogram the spine head volume, what we see is that the best fitting model is not a continuous, a graded analog distribution, which is that reference shown in blue. The best fitting model is actually a sum of a binary variable and an analog variable that's drawn from a log normal distribution. Now, there are a variety of learning schemes that could give rise to this distribution. There might be multiple distinct learning rules at work here. But the key is, now we know, now we have the explicit constraint from biology that this is how biology is able to achieve energy efficiency, using whatever learning scheme gives rise to this distribution. And this can provide guidance for future algorithm development work. Another fundamental question we would like to answer in these data is, what is the minimal unit of computation in the brain? If you think about how neurons are modeled in deep neural networks and in neuromorphic hardware, typically they use a single compartment model that takes a weighted sum of its synaptic inputs, applies a nonlinearity, and then sends the signal off to another location. By contrast, we know that neurons in the brain have very complex morphology, and they receive synapses onto every part of their morphology. And so our partners at the Allen Institute did a census of all the different inhibitory cell types in the brain and where they synapse onto excitatory cells. And so these vertical bars in this slide are histograms of which compartment inhibitory neurons synapse onto on an excitatory cell. And if you just look at the green bar, that is one type of inhibitory cell called a chandelier cell that only synapses onto the axon initial segment of an excitatory cell. Its one job, and I'll hop out of your way so you can see, its one job is to turn off the outputs from excitatory cells so that it can't send messages anywhere else. There are other categories of inhibitory cells that preferentially target the apical dendritic compartment or the basal dendritic compartment close to the cell body. So we are now able to precisely define the circuit motifs and to constrain the space of computations that might be performed by single neurons in the brain. Perhaps that can guide us toward minimal models of neurons in algorithms and in neuromorphic hardware so that we can capture the brain's computational complexity without needing to move as much data around on the chips. One other entitlement of Micron's data is we are able to study long-range interactions, feed-forward and feedback interactions between different regions of the brain. Now, one motif that you see in deep neural networks is all-to-all connectivity between layers. That is not what we see in the brain. In fact, we see quite sparse connectivity. And we are actively engaged in studying the relative tuning properties of neurons that interact over long distances to understand what is the principle that determines whether two neurons should be connected over long distances. Now, I'm not going to preempt forthcoming papers by our researchers, but you can expect to see a lot of exciting insights coming from this analysis in the future. Just to conclude this portion of the talk, historically, the field of machine learning has progressed through the work of brilliant people and lots of trial and error building models. And this has gotten us pretty far. But it's clear that a paradigm shift is needed to deliver models and hardware that approach the performance of the human brain. IARPA determined that scaling up our window onto neural circuit structure and function could transform our understanding of computation in the brain. And now we have provided the neuroscience and machine learning communities with the resources that everyone can use to gain clear guidance for future algorithm development. As with our work on data storage, this was science fiction just a few years ago. And now we are seeing a steady march of new algorithmic guidance coming from Micron's data, which will continue for the next decade. In conclusion, biology really is all you need. We've worked very hard at IARPA to understand where we can get guidance for solving some of our most pressing scalability challenges in storage and computing. And once we determined that biology had already solved these problems, the challenge was just building bridges between biology and engineering disciplines so that they could take advantage of this insight. I've worked very hard throughout the last 8 years that I've spent at IARPA developing new technologies and cultivating communities that bridge biology, engineering, and computer science. And I'm very excited to see the real-world impact that this work is going to have in the future. Thank you.
Very exciting talk. Any questions from audience? As you line up, there's a question came online for your first part of the talk, which says, are there any special conditions needed to preserve the accurate or the error rates low for the DNA, and what's the cost implications of that?
Are there any special steps needed to?
Or special conditions to keep the DNA so that it doesn't degrade?
Yes, yes. DNA is very susceptible to light.
I see.
And to temperature. So it's important that for storage applications, DNA be protected in a relatively dark environment that is not caustic. But there are several techniques that have been determined that can allow us to preserve it for long periods. You can encapsulate DNA in silica beads. You can store them in a box, right? So there's a variety of simple and complex solutions.
Cool, thanks.
Thank you.
Go ahead.
Can you tell us a little bit more about cooperation, international cooperation, and especially with the European flagship project, Human Brain Project?
I think the question was, can I talk a little bit more about international collaboration and the connections with the Human Brain Project, right?
Yeah.
So, IARPA funds work internationally. We want the best people working on the hardest problems wherever they are found. The Microns program made contract awards for the work that I just described to institutions in the United States, but excuse me, but there were researchers who participated from Germany and from a couple of other places on the machine learning side. And so we have benefited tremendously from researchers in the EU. There were no explicit links between the Microns program and the Human Brain Project. The focus of HBP was to take all of our existing knowledge about the brain from the existing experimental literature and then build a complete model of cortex, of much of the brain as possible using those constraints. Microns is taking a different approach. We're starting with the premise that we don't have enough experimental constraints, and we've gone and developed the technologies and acquired the data needed to achieve that.
Thanks, Doug.
Thank you.
Very exciting talk. So this is Qipeng from NC State. So I'm sure that we can learn a lot from nature, from brain in this case, from biology. But the one thing which already shown is modern computing actually can achieve lots better results than what our brain can do. For example, we now have AlphaGo, which actually can beat the top players in the Go, who actually get trained the mean of the whole life. But here, AlphaGo actually got trained just in short period of time, much shorter period of time. So I'd like to, basically, I think there's some kind of potential synergy, I would think, between the traditional computing or the modeling and the brain modeling. But I want to hear about your comments about this. Thanks.
Thank you.
So the question was, are there opportunities for synergy between brain-informed computing, as I've described it here today, and traditional computing, which, no question, has achieved amazing advances, right, over the last several decades? And the short answer is yes. Of course, the key to the power efficiency of the brain, we think, is that it localizes storage and compute and only moves data around when it's absolutely necessary. Your typical von Neumann computing architecture moves a lot of data around. And so I would love to see a dialogue between the neuromorphic computing community and the traditional HPC community to understand how we can blend the best of both worlds to achieve power efficiency for specific applications like AI, while also leveraging some of the advantages of traditional compute. Thanks.
Thanks. Go ahead.
Yeah, and my question is similar to Xipeng's comment. So I think if the title is correct, then the assumption that biology already found the ultimate optimal solution in terms of intelligence. But as we know, we have a lot of recognition and biases in our brain. If following whatever our brain is using, we may copy the same bias, all those bugs in our logic or function of our brain. So what's your comment about those things? Thank you.
Could you repeat the question?
I can't understand. Sorry.
Okay. Our brains have a lot of kind of flaws, like biases. If we
So he's saying we have all sorts of flaws, for example, in either disease-related or how can this hybrid interaction can improve the human brains in the long run.
Yeah, nobody wants to build a neuromorphic computer that has inbuilt disease, then you wind up with HAL.
Oh, I did not know disease. Just normal people, we have a lot of flaws in our logic, in our brains. So we have like, I think if you look up Wikipedia, we have like 100 biases in our brain. So how to counter those kind of things? Thank you.
Thank you for the question. I think the question was, there are a lot of flaws in the human brain. Biases, things that we are trained to do inappropriately, mistakes that we make, and how can you avoid building that into AI systems? There is an entire field of algorithmic bias in AI, which deals with exactly that issue, right? It's a problem in AI today. If you have data that capture biases of society, then you get AI systems that are flawed in the same ways that people are and can produce malicious content. So I don't think this problem is specific to neuroscience-informed AI. I think that this is a machine learning problem. But I agree with you that we definitely need to be mindful of any biases that are specific to the operating principles of brains and what that might lead to in the future.
Thanks,
I don't think that that concern is any greater than algorithmic bias in AI, but we should be sensitive to it.
Go ahead.
Right. First, how much metadata infrastructure proportionally would it take to know what's inside your desktop DNA storage device? Second, your early graphic on storage mediums and the different comparisons there, how would that graphic be affected by the codec encoding or error correcting that's going on underneath the hood? And third, do you appreciate the irony of asking for a three-dimensional storage medium and then giving a one-dimensional solution?
It's okay. Answer the first question.
Okay, so there were two technical questions and then one. Well, let's talk about your first two questions.
Just
okay.
The last one's just for fun.
Okay.
Yeah.
So the first question was, how much metadata is required? So it is codec dependent, and it also depends on how you plan to access the data. If you intend to just do a complete bulk retrieval of all your data in an archive, then the amount of metadata you need to store might be less, because you just need to store an address that shows where the data block is in the archive at large. If you want to be able to do content-based retrieval, random access from the archive, you're going to need more metadata that's encoded in each oligomer so that you can selectively pull that out. But I do think that the amount of metadata that is used is ultimately tied to the codec that you're using. I know that's a kind of a vague response, but it is true. I believe the second question, I've forgotten the second.
I didn't remember either, sorry. That's okay. Let's have the last question.
Thank you, though.
Thanks.
Hi, thank you for the presentation. One quick question. So I'm Razvan Marinescu from UC Santa Cruz.
Okay.
The question is, you spoke about storage and compute and how biology can inform these. Are there any other problems where we can get inspiration from biology apart from these two?
Short answer.
Yes, and I would love to discuss them with you after I get the hook off the stage. I personally focus on the interface between biology and computing, but there are a large number of problems in chemistry and in the synthesis of chemicals that biology has devised very clever solutions to. So we can look at the molecular scale, also the way that genes are regulated inside cells. It's kind of a fuzzy logic, I think can be very useful for control. And I'll leave it at that. Thanks.
Thank you.
Thanks, Dr. Markowitz.
Thank you, everyone.